Ladies and gentlemen, welcome. It's a pleasure to be here. And my name's Richard Emsley, the Executive Dean of the Institute of Psychiatry, Psychology and Neuroscience, and it's a real pleasure to welcome you here to what is a celebration of the fantastic work that goes on in this institute. It's great to be at something where we celebrate rather than being at an event where I'm trying to sort out a problem. So that's great for me. I just want to remind you that the idea for this event was started a number of years ago by Till Wykes and then was taken over by Talia and Carmina as they chaired the Research Innovation Committee. And what we want to celebrate this afternoon is the breadth and depth of research that goes on at this institute, from social science to molecular sciences, and it's about celebrating the diversity from very junior people up to full professors. It's all about celebrating diversity within the institute, and it's about promoting collaboration and networking. So I've been told quite strongly by Carmina to say to you, you must go to the coffee, and you must mingle, chat and network with one another. With that, I want to start off now by, if Carmina can help me, by presenting the Independent Research Awards for 2017 and 2018. And the first Independent Research Award of the Institute goes to Dr. Alicia Hughes from the Department of Psychological Medicine. Yes. Thank you.
Thank you.
Alicia, I will do a picture of the gathering in a second. So
yes.
Let's wait on that.
Sorry. And now it gives me a pleasure to ask Dr. Kim Chisholm from the Wolfson Centre for Age-Related Disease. Right. And then the next one for the Independent Research Award from the NIHR Maudsley Biomedical Research Centre is Dr. Billy Gazzard from the Department of Psychological Medicine. And then also for the NIHR Maudsley Biomedical Research Centre, Dr. Sadie Boniface from the Department of Addictions. Well done. So from April 2018, the second round of the awards this year, again for the two independent research awards from the IoPPN. The first one goes to Dr. Douglas Lopez from the Wolfson Centre for Age-Related Disease. And then next, Dr. Susanna Roberts from the Department of Psychology. And then from the April 2018 NIHR Maudsley Biomedical Research Centre Award, the first goes to Dr. Karen Hodgson from SGDP. Then last but not least, the NIHR Maudsley Biomedical Research Centre for April 2018 goes to Dr. Eleanor Makovac, Department of Neuroimaging. So we're just going to have a quick group photo, then I'll come back. So it now gives me pleasure to hand over to Carmina, who's going to chair the first session. And I just want to let you know earlier on, there will be a pause at some point in the session where we'll have a minute's silence to mark the first anniversary of the Manchester bombing. But you will announce when that's to occur. So please help me welcome Carmina to the stage.
Thanks, Ian, and thanks all of you for being here today. I want to thank Talia, who's the co-chair of the RIC, with me, who will chair the second half of the session. And as Ian said, please do mingle around, stay as long as you can, come back for coffee breaks and drinks if you can. Now, the first speakers, for both the first session and the second session, the first speakers are always speakers that come from the broader professional services family, because we think that the IoP star, the IoPPN stars are not only, of course, the academic and the lecturers and the researchers, but also the people that they create the infrastructure that allow us to do all the work. So the first speaker is Peter Bishai from the CRF, the Clinical Research Facility at King's College Hospital. Peter.
Hello, thank you for inviting me to speak to you today. I'm going to talk to you about the NIHR Wellcome Trust King's Clinical Research Facility, which was formally opened by Dame Sally Davis, the Chief Medical Officer, in May 2014. The unit comprises two main research facilities within King's College Hospital, a 3-floor standalone new build, and the clinical trials facility. The facility was part funded with a capital award from the Wellcome and additional awards from its academic health science partners, King's College London and the NHS Foundation Trust of South London and Maudsley, Guy's and St Thomas's and King's College, along with a number of private benefactors. Each area within the facility was designed and built to complement the other in terms of ability to offer a unique and neutral space to carry out clinical trials in a dedicated research setting. We're keen to facilitate the work of established investigators, encourage and support the use of the new facilities by new investigators, providing an environment with which the pharmaceutical industry is able to collaborate actively with our clinical teams. We can accommodate all types of trials, ranging from day and overnight studies to longer-term stays. Our focus is particularly on experimental translational early phase trials. We specialize in mental health and neurological studies, hemato-oncology, endocrinology, cardiovascular and respiratory medicine. All are supported by a team of nurses and technical staff. Later phase studies can be brought to the clinical trials facility, but nurse support cannot be provided for those. So the experimental medicine facility is housed over 2 floors within the main build of the CRF. Entry to both floors of the EMF is via the main hospital corridors at KCH, and it should be noted that this physical and intellectual link to the hospital and all the support systems it offers are critical to our operation. The first floor provides the main entrance and reception area with easy access to 4 interview rooms with video recording facilities, 2 clinical rooms, each with an examination couch, and an open plan area with 6 reclining chairs for sampling and monitoring study subjects. Additionally, there are 2 state-of-the-art electronically and acoustically shielded rooms ideal for monitoring and recording brain, heart, and muscle electrical activity. There is also a purpose-built virtual reality suite fitted with a sophisticated integrated computer-based system providing an environment for real-time assessment of thoughts, mood and symptoms as they occur in psychological studies. The ground floor of the EMF includes a 2-bedded intensive care ward for the study of all critically ill patients who are ventilated or have suffered stroke, trauma or brain injury. There is also a 4-bedded area for experimental medicine studies with capacity to closely monitor aspects of study subject cardiovascular and respiratory physiology. In addition, there is a newer imaging suite comprising a 3T functional MRI scanner along with associated support rooms. Also on the ground floor is a standalone facility for commercial trials. The clinical trials facility falls under the umbrella of the CRF and has been designed in conjunction with current users and industrial collaborators to provide a state-of-the-art facility for commercially sponsored clinical trials. The CTF, as it is known, divides into 2 major areas: an 8-bedded facility including 1 isolation bedroom, an area supporting 11 fully reclining chairs, and both areas are open plan with easy access to private individual consulting rooms where study subjects can be interviewed and examined appropriately and confidentially. On the 3rd floor of the CRF is the cell therapy unit. It's the largest of its kind within an academic environment in Europe, not in terms of meters squared, but in the fact that it has the capacity to produce 6 final products simultaneously within its 20 rooms operating to air qualities A, B, and D. The unit was specifically designed for multi-product processing, having been built under a rigorous risk analysis process approved by the Medicines and Healthcare Products Regulatory Agency. The unit provides good manufacturing practice facilities licensed by the MHRA and the Human Tissue Authority for producing autologous and allogeneic cell and gene therapy products for clinical trials and routine therapies. The shared facility supports research from King's Health partners, including bone marrow transplant, mesenchymal stem cell production, cell and gene-based therapies, and work from the Institute of Liver Studies and the Department of Diabetes. Thank you.
One more, okay, 1 second. I just want to tell all the speakers that Abby will be the timekeeper. So she has a 1 minute, and then she has a red one. End of time. Perfect. And then before we start, I just want to thank Peter. For those of you who recently started to work at the IoPPN, you just don't know how difficult life was before we had the CRF. So, you know, you give this for granted now, but actually has made a complete difference in the work of people like me and others that do experimental medicine. So thank you very much. And now, Professor Richard Emsley, Professor of Medical Statistics and Trial Methodology. Richard.
So the randomized trial is still seen as the gold standard way of seeing if an experimental or new treatment is better than some control. And the MRC trial of streptomycin in 1948 is still widely considered to be the first fully reported randomized trial. And its design has become the leading clinical trial design over the past 70 years. This is a standard 2-arm parallel group trial where patients are recruited and randomized into 2 different groups and then followed up for the duration of the study. A review in 2014 showed that about 80% of registered trials still follow this 2-arm parallel group design, but around half of definitive trials fail to show a significant benefit of the new treatment over the comparator. So my question is, why do we continue to persist with this 2-arm parallel group design? And my pitch is that in the 21st century, patients and the NHS need more efficient randomized trials, especially in mental health. In our psychosis therapy trials here at the Institute, such as Avatar and Slow-Mo, we're improving on this, and we're using more active control arms which match for contact time which is delivered in the therapy, or considering different choices for treatment as usual. With digital interventions, we need to make sure that the technology is relevant to the end of the trial rather than just being fixed at the start of the study when we begin the evaluation. Ultimately, what we want to know is, can we deliver quicker results for patients? Can we speed up the development of new treatments and improve our success rate? Can we answer more questions to improve trial analysis? The primary analysis of any randomized trial is based on the intention to treat principle. So we analyze patients according to their randomized arms. Our biostats department here at the Institute and my previous research group in Manchester have led the development of statistical methods to think about and answer more interesting questions about treatments. We want to know, who does the treatment work for? Can we identify and test whether different subgroups of patients benefit most or least? And ultimately, can we get to the point where we personalize treatments to that individual and test that within a trial? What happens if you actually receive the treatment as opposed to if you're just offered the treatment? Where's the optimal place for a patient to receive the treatment? When is the optimal time within a patient's condition to receive that treatment? Why is the delivery of different treatments why is the delivery of treatment more effective in some contexts than in others? And how does the treatment work? So what happens after that randomization, taking into account things like compliance and what the active mechanisms are by which a treatment will bring about a change in clinical outcome? And there's lots of challenges with that analysis, including unmeasured confounding, even in randomized trials, and accounting for the effects of different therapists. We summarized 10 years of our MRC-funded methodology research in this open access monograph, which is intended for a non-statistical audience, and it shows that mental health is actually ahead of many other disciplines in thinking about these analysis issues. Where other fields, and especially cancer, have moved forward is in trial design, and I'm going to highlight 3 different trial design options here. So in a multi-arm, multi-stage trial, several treatments are simultaneously assessed against a common control group within a single randomized trial. The multi-stage aspect allows us to drop arms which are not showing benefit in a pre-planned interim analysis. So here, treatment 3 is dropped after phase 2, and treatment 2 would be dropped after a subsequent interim analysis during phase 3. This is an example of an adaptive design, a seamless phase 2, phase 3 type design. It's more efficient than a series of corresponding parallel arm group designs would be, and we can add in new treatment arms as new comparators become available, enabling new comparisons. So these are often referred to as perpetual trials because they never need to end. The example here is the MRC Stampede trial in prostate cancer, which has now recruited over 10,000 patients and is going to answer 11 different questions about treatments in 18 years that would have taken over 50 years to answer in the parallel group approach. A second approach is called smart designs, and the key feature of smart trials is that for patients who don't respond to the initial therapy, regardless of which that initial randomization is, they're re-randomized to receive either a supplement of that initial therapy or they switch to the next choice of the first-line therapy. And the aim here is to test the optimal individual patient pathways in chronic disease conditions. In menu trials, we can remove diagnostic criteria and we focus on the common underlying mechanisms across disorders. So we screen patients for their dominant mechanism, and then we enter them into a trial where the treatment is particularly focused on that mechanism. In cancer, these mechanisms are typically genetic markers of the tumour, but we can extend this thinking to neuroimaging markers and psychological mechanisms. And ultimately, we want the IoPPN to be leading the way for efficient trials in mental health. Thank you.
Thanks. Sarah Mizielinska, lecturer in dementia and neurodegenerative disease. Sarah.
So I'm going to talk to you all the way, so we're going to switch from clinical trials actually all the way down to molecular mechanisms, and actually from biology almost to physics, but I'm not a physicist, so don't worry. Great, thank you. So what I'm going to talk to you today is about a concept in physics called phase transitioning and the relevance of this to biology and actually neurodegenerative diseases. And it's a concept that we can see through our everyday life through things like this photo. So this photo is a mixture of oil and balsamic vinegar, which I like personally. And what you can see is, though, both of these substrates are a liquid, they don't just mix into a light brown solution, they actually separate. And so what we can see here is the balsamic vinegar is going from a soluble form actually to a liquid droplet. So as you can see, these droplets here. And actually, different components can actually transition, going from actually a liquid to more like a gel-like state and actually onto a solid-like state. And this is becoming really important in the organization of the cell in biology. And actually it's thought that this is the organization of primordial life, where things are being brought together for an advantageous purpose. And you can see this if like an enzyme and a substrate are brought together into close proximity, obviously they're going to be more effective. So when we look at a cell, we can see many examples of these lipid droplets, and they form what's known as a membranous organelle. So this is just a structure with a very specific function that is not enclosed by a membrane. And so there's many different processes going on here. Lots of them involve nucleic acids of some form. And of my interest to me, it seems that proteins that are associated with neurodegeneration, particularly dementia and motor neuron diseases, actually are involved and normally found within these structures. And then they become changed upon diseases. And actually several mutations in these proteins actually cause changes to these structures. And I can give you an example of this here. So this is a slice of brain of a person with dementia. And so the brown stain here is a nucleus, and so this is where the protein is normally found. And stress in the cell, stimulates its moving from the nucleus to the cytoplasm. And it's thought that quite often these move to stress granules. This is a liquid body involved in stress. And then they accumulate, and it goes from being a liquid structure to a more solid-like structure, and then eventually to a more fibril-like structure, which forms these really giant aggregates within a neuron of the brain. And these aggregates actually sequester lots and lots of protein from within the cell. So you actually get complete clearance of this protein from where it normally resides in the nucleus. And of course, if it's not within the nucleus where it's normally functioning, it can no longer carry its normal function. And this is just an example of a study I've just completed, that they're not separate bodies, they're not individual proteins within an individual component. They actually overlap quite a lot. And so here I showed that the nucleolus, which is here, which is the site of ribosome assembly and therefore all protein translation, is dramatically affected by the presence of these cytoplasmic inclusions in a type of dementia called frontotemporal dementia. So, and my current studies are really focusing on the nuclear pore complexes. And as I said, the initiating process in a lot of these is when things move from the nucleus to the protein, and obviously they move through this nuclear pore complex. And you can see here, the interesting point about this is that they form a hydrogel in the inner structures. So we've actually been forming these gel-like structures in vitro, and to try and see how the proteins associated with dementia can affect the phase transitioning of these nuclear pore selection barriers. And then also using super-resolution microscopy to identify individual nuclear pores and look at transport through these pores. And so we can track the movement of these through the hydrogels and then link these to common disease pathologies. And we're hoping that the insight into these very precise molecular mechanisms will give us new targets for disease. And this is a program that's funded by the UK Dementia Research Institute, which is a big collaboration, big funding from the government, stimulated by the MRC, Alzheimer's Society, and Alzheimer's Research UK. Thank you very much.
Tom Freeman from the Department of Addiction. Tom?
So thanks very much for the invitation to speak today. It's a pleasure to be here, and I'm going to talk to you about cannabinoids, mental health, and addiction. And really, cannabis has become a major problem in addiction services in Europe. So you can see this on the left panel here. So cannabis, shown in red, has superseded opioids as a major reason for coming forward for treatment for the first time. This is really unprecedented, and it raises significant problems for treatment providers. And one of the things I've been interested in is whether cannabis products might have something to do with this. And we've heard a lot about cannabis potency changing over the years. And the cannabis plant produces over 100 naturally occurring cannabinoids. The main psychoactive constituent is THC, delta-9 tetrahydrocannabinol. Some cannabis products also contain cannabidiol or CBD. CBD is non-intoxicating and potentially protective. And so the best place to go if you're interested in cannabis products in Europe is to Lisbon, where the European Monitoring Centre for Drugs and Drug Addiction is. And this is where I visited last month as part of my fellowship. And I analysed data across Europe, and you can see this on the right panel here. So you can see that across Europe, cannabis increased over 11 years from roughly 5% to 10%, so roughly doubling during this 11-year period. But of course, Europe is a very big place, and it helps to focus on one region to look at these associations. And if you're interested in cannabis, really another good place to go to is the Netherlands. It really is. And the reason I chose this place is not because of the coffee shops, it's because of the sampling methods they use. So because cannabis sales are tolerated, they actually have randomized sampling from coffee shops, similar to trading standards for other products in the UK. This is much better than police seizures that may be biased. And so you can see here changes over 16 years in THC shown in red. It's a very interesting profile of change, characterized by an initial increase followed by a subsequent decrease. And then looking at first-time entry to addiction services shown in blue, you can see that these also show a similar profile of change with a time lag of a substantial increase, very, very big increase in treatment demand, followed by a decrease. And so this association persisted after adjusting for age, sex, and non-cannabis treatment admissions. In another study I looked in the UK, closer to home, and this was looking at different types of cannabis, which you may be familiar with, a high-potency herbal form here, and a low-potency resin or hash here. And I found that the high-potency forms of cannabis were linked to greater adverse outcomes, including cannabis dependency, memory impairment, and paranoia. And again, these associations persisted after adjusting for confounding variables. But one limitation of this approach is that this form of cannabis has high THC, no CBD, whereas this form of cannabis has balanced THC and balanced CBD. And we know from recent police seizure data that this kind of cannabis is very rare in the UK nowadays, only 6% of the market in the UK. So if you want to look at the effects of CBD, you really need to take these compounds to the laboratory. And so in the lab, I've conducted several studies using psychopharmacology as an approach. And in these studies, we typically administer placebo alongside THC, a single dose, the same dose of THC combined with CBD. And what we've shown over the years is that CBD can be protective across multiple domains. These include cognition, so verbal memory. It's a very interesting finding that's been replicated. Also psychosis and psychotic-like symptoms. This is very relevant because CBD has shown promise as a treatment for psychosis in clinical trials, one of which was based at the IoP. Also addiction, we've shown across cannabis and tobacco use disorders that CBD can reduce the salience of drug use. And more recently, cardiovascular effects. So this was a recent study where you can see placebo in black produced no change in diastolic blood pressure. THC produced an increase, shown in red, and this increase was blunted when it was combined with CBD. And so really what the implications of these findings are is that cannabis can be made safer by reducing THC and or increasing CBD. And this is something that the Liberal Democrats proposed in their election manifesto, which wasn't very successful for their vote winning, but it's something that in Canada they're trying to implement, and worldwide we might see more of this. And the real gap in the knowledge is which ratio of CBD to THC is most effective. And ongoing work at the IoP, myself, Amber England and colleagues is trying to find out which ratio is most effective for reducing harms. And in other work, I'm also testing whether CBD can be effective as a treatment for cannabis use disorder in an adaptive randomized clinical trial. Thanks very much.
And now Helen Fisher, Senior Lecturer and MQ Fellow at the SGDP. Helen.
Great, so thanks again for the opportunity to be here. The aspect of my research I wanted to share with you today focuses on psychotic symptoms. So when I talk about those, I'm thinking about people who are hearing voices, seeing things that other people don't see, perhaps being extremely paranoid and having other unusual beliefs. And we normally think about these symptoms in relation to disorders such as schizophrenia, but actually we know that they're surprisingly common in the general population as well, particularly amongst children. So if we were to take an average class of 12-year-olds, for instance, we might expect one, if not two, of the kids in that class to be currently experiencing psychotic phenomena. And perhaps not surprisingly, these types of symptoms are quite distressing for the child, not always, but most of the time. And they're often associated with other problems at the same time, so the kid might also be self-harming. But they're also an important early indicator that these kids are likely to be at risk for a range of severe mental health problems later, so including schizophrenia, but also post-traumatic stress disorder and anxiety disorders, and also that they're likely to have other difficulties in early adulthood through to mid-adulthood at least, in terms of certainly attempting suicide, struggling in terms of being more likely to be lonely and socially isolated, having a child at a much earlier age, and generally having a poorer quality of life. So therefore, it's really important to understand which children are likely to go on and have these particular symptoms, and what we might be able to do to protect them from developing them. And that would really allow us to start to develop interventions to target at the most vulnerable kids to really improve their quality of life. So that's what some of my research does, and really to address this problem, what we've been very lucky to have access to is data from the Environmental Risk Longitudinal Twin Study, or ERISC for short. And that's a study where they've been tracking the development of 2,232 twins who were born in 1994 and 1995 across England and Wales. And those kids and their families have basically had literally every aspect of their lives assessed at multiple times in childhood and adolescence. And we're really thrilled that 93% of those kids came back and saw us when they were 18 to be assessed again. And that's really important because it means we've retained most of the kids we're really interested in this sample. So using that fantastic data, what my research team have been able to do is show a variety of things. So we've seen that kids who are exposed to various forms of victimization in childhood, so whether those be bullying by peers or physical abuse from parents, We've shown that alterations in how their genes are operating, and also that if they grew up in an area that has poor social connectedness or high levels of air pollution, that those things are much more common amongst kids that will go on and develop these psychotic symptoms. And that really starts to give us a bit of a clue as to which are the kids that might be most in need of our help. On the converse, though, it's also really interesting to think about the kids who have all these risk factors but don't go on and develop psychotic symptoms. And really we can learn about what protects them to really help us to inform the content of these preventive interventions. So this is just some work from one of my PhD students, Eloise Crush, who's shown that having better cognitive functioning, living in a family which is happier and more stimulating in terms of its environment, and living in a more socially cohesive community all protected children from developing these symptoms, even in the context of being exposed to multiple forms of victimization. So ultimately, what our research is hoping to do is really to inform the development of interventions that can be targeted at the most vulnerable children and ensure that they can get the right support as early as possible. And hopefully, therefore, we can actually prevent the development of these psychotic symptoms in some of these kids at least, and hopefully eventually prevent the development of severe mental illnesses in a good proportion of these kids in the population, and hopefully help more young people to lead happier and more fulfilling lives. So I will just finish there, and to thank both MQ Mental Health Charity and the Medical Research Council for their very generous support of lots of different aspects of this work, and particularly to the kids and their families who really have let us invade all of their lives for 18 years, and to all the research team, the staff and the students involved in analysing, collecting all of this data. And thank you for your attention.
Now we have Nuria Marcus, who is the first of the PhD students presenting. All the PhD students presenting at the Parade of Stars are the final, those who get to the final of the 3 minutes competition. So in fact, are the only one that have a ex-official role at the Parade of Stars. So, Nuria.
Thank you. Hi, my name is Nuria, and I'm a third-year PhD student at the Department of Child and Adolescent Psychiatry. And for my PhD, I want to examine how early experiences shape the brain. With early, I mean the first few years of our lives. And more specifically, I'm interested in what happens if these early experiences are adverse, like abuse, neglect, trauma. So let's go back in time and have a look at the background of our study. Before the 1990s in Romania, many children were living in orphanages under the Ceausescu regime. There, most were suffering under extremely harsh conditions. There wasn't enough food, no toys, no permanent caretakers. In some orphanages, less than 50% survived the winter. After the fall of the regime, many of these children were adopted, bringing about a very sudden change of their circumstances. They were now living in very nurturing and loving families. The English and Romanian adoptee study follows the development of some of these children who were adopted into UK families. As a comparison group, the adoptee study also follows some children who were adopted within the UK and within the first few months of their lives. So they had the same adoption experience, but without the extreme adversity of the Romanian orphanages. What the adoptee study has found so far is that while some of the Romanian adoptees grew up to be fine, with quickly catching up and with developmental outcomes comparable to the UK adoptees, many of those who were in the orphanages for a long time, specifically longer than 6 months, have developed a persisting pattern of problems. This pattern includes symptoms of ADHD, autism, and disinhibited social engagement, which is the inappropriate, overly familiar behavior towards strangers. Now that the adoptees are in their mid-20s, for the first time, this study also includes scanning our participant brains in an MRI scanner. And this is where my PhD comes in. My PhD is the first to examine how these early experiences impact on adult brain structure. I want to find out whether this extreme adversity early in life has left scarce in the brain despite the nurturing upbringing afterwards. So to do this, I have used the software FreeSurfer to automatically segment the brain into its different regions and to calculate values for cortical thickness, surface area, and volume across the whole cortex for every participant. I then compared the UK adoptees and the Romanian adoptees to see if there's any differences between these two groups in these measures. And also within the Romanian adoptees, had a look if there's a linear relationship between these measures and the amount of time that they spend in these orphanages. What I have found so far is that the Romanian adoptees have significantly smaller brain volumes compared to the UK adoptees. And also, the longer they were in these orphanages, the smaller their brains are, even today. Moreover, the surface area and volume of a region in the frontal part of the brain that is involved in impulse control and motivation is also directly related to how long the adoptees were in the orphanages. So what we can see here is that if we control for the overall brain stunting that we see, this region seems to be positively related to the amount of time that they were in the orphanages. So while overall the brain gets smaller, this region seems to be relatively less affected by that stunting. So this shows that even more than 20 years after it has ended, early adversity has an impact on brain structure, and it affects some regions more than others. So what's next for my PhD? I hope that I will be able to link some of these changes that we see to the pattern of symptoms that we often see in those who have been in the orphanages for a long time. So, for example, what we find is that if you look at the Romanian adoptees who were institutionalized for more than 6 months, the total brain volume is negatively related to the number of symptoms of disinhibited social engagement. So the smaller their brains are, the more symptoms they have today. I hope that this research will help to gain a better understanding of the consequences of early adversity, and in the long term, a better understanding of the mechanisms that make the first few years of life so important for brain development. If you want to know more about our research, follow our updates on Twitter. And I'd also like to thank my supervisors, Mitchell and Edmund and Graham, for the amazing support, as well as Dennis and Sagari, who also work on the study and focus on the functional MRI and the DTI aspects of this project. Thank you.
And now Professor Elizabeth, or Liz Bradbury, Professor of Regenerative Medicine and Neuroplasticity at the Wolfson Center.
Thank you. So my lab is interested in restoring function after spinal cord injury. So we've got two main research questions in my lab. Firstly, we really want to understand why the spinal cord does not repair itself, unlike other tissues of the body. And also, how can we try and develop regenerative therapies aimed at restoring meaningful function after spinal cord injury? So why is spinal cord injury such a big problem? Well, there are currently 2.5 people worldwide living with a spinal cord injury. But as a medical problem, it's dramatically increasing. There are 130,000 new cases every year. Spinal injury patients now live a near normal life expectancy, but often with these lifelong disabilities, and sometimes needing high dependency care for the rest of their lives. There are also no adequate therapies, so the only thing offered to spinal injury patients at the moment are rehabilitation or treatments for secondary complications. So this is really a major medical problem, and we really drastically need some new regenerative therapies. So to develop these therapies, we first need to understand the basic biology of the injured spinal cord. So we need to understand some of the cellular and molecular changes that happen after a spinal cord injury and lead to this really dramatic pathology that does not resolve. Things like the invasion of these aggressive inflammatory cells. And it's very important to also use clinically relevant animal models, so models that mimic the pathology that you see with a human spinal cord injury. So once we can understand the pathology a bit more, then we can start to develop regenerative therapies. These are typically aimed at enhancing axonal regrowth, or what we call neuroplasticity. And here's some examples here in the spinal cord of experimental animals that have undergone some neuroplasticity regenerative therapies experimentally. It's also really, really important to decide what kind of functions you want to restore, what you're aiming to restore with these regenerative therapies. And we really want to target functions that are the highest priority for spinal injury patients. So the majority of clinical spinal cord injuries occur at the cervical level, the neck level, and they affect all 4 limbs. So recovery of hand function is the highest priority for these patients, and that would enable them to do everyday activities such as just picking up a toothbrush, being able to write, being able to hold and lift a drink. So these are things we take for granted, but they actually involve these really complex sensory and motor integration, which all happens in the spinal cord, and that's lost after a spinal cord injury. We try to model these important functions in experimental animals in my lab, and these are rats that are actually reaching and grasping for sugar pellets, and animals that are crossing horizontal ladders. So to do these behaviors, they need to be able to grasp the rungs of the ladder and then use motor coordinated movement to get across. So they're really complex tasks. Things that we think are really easy, they're actually very complex when you think about them. So this is kind of what we're trying to address, the key questions, and I'll just show you really quickly a couple of examples of how we're addressing that in my lab. So as I said, it's important to have an experimental model that mimics what you see with a human spinal cord injury. And we think the rat contusion model really effectively does this. And typically after a human spinal cord injury, the damage gets worse over time. We have what's called a secondary injury pathology. And you can see this here in our animal model, in our adult rats. Over time, the damage gets worse. You've got a loss of more spinal neurons are being lost. You get these holes or cavities forming in the lesion site, and you get this reactive glial scar that forms around the injury. And it's very similar to the pathology you see with a human spinal cord. So this is a human pathology plate, and you can see this is the typical chronic pathology of a spinal cord injury. A huge hole in the center of the spinal cord, surrounded by this dense scar tissue. And we know from many years of research, both in my lab and many other labs, that it's this spinal injury scar that really blocks regeneration and new growth. So this is why we're trying to use therapies that break down some of this scar tissue or some of the inhibitory components, the inhibitory matrix of the glial scar. And we use things like enzymes such as chondroitinase. If you inject chondroitinase into rats, you get this really significant enhancement of synaptic activity. And you can see here that synaptic activity is lost after spinal cord injury, but recovered after chondroitinase treatment. And we're actually using a gene therapy approach because we think gene therapy, delivering this enzyme with a gene therapy approach, you get a much more optimal delivery to the spinal cord. And finally, just to show you our very latest work with a novel chondroitinase gene therapy approach. So this gene therapy is engineered, the viral vector is engineered with a gene switch, so we can actually control gene expression. We could switch the gene on and off simply by giving doxycycline. In this case, we give doxycycline in the animal's drinking water. If we take away doxycycline, the gene switches off. So this way we can look at short-term versus long-term delivery. We've compared 2 weeks versus 2 months. And really excitingly, this is our latest finding, by giving the gene, when the gene is long-term in the on position, you get this really nice recovery of skilled reaching in adult rats that have had these cervical level injuries that affect the upper limb and hands. And just to show you why we think these results are really exciting and potentially could have a really big impact, is this just shows the hand movements of rats compared to humans. They use very similar coordinated movements to reach and grasp for objects. And so that's why we think it's really important and hopefully going to be able to be clinically developed. And since this is the parade of stars, I want to highlight the real stars of all this work, which is my amazing lab who have done all the hard work for these projects. Thank you.
So can I invite everybody now to just hold a minute of silence to remember the victims of the Manchester Arena attack last year, please. Thank you. So we'll start again now with my good friend, Professor Craig Morgan. that hopefully will not be disturbed so much by the noise. If it does, please, we have a few minutes earlier, so if it becomes unbearable, we'll stop and then go and check and start again. Great.
That's okay. Thanks very much, Carmine. Coincidentally, I want to start with another tragic event, an event that occurred in the early part of the 20th century. And what this slide shows you is the proportions who survived by an indicator of social class. Anybody, any ideas what this event was? Yeah, this was a sinking of the Titanic. And it's not our work, but it's an illustration of the kind of work that we do. This is an illustration of what we mean by social epidemiology, trying to understand the social distribution and determinants of health outcomes over time. And by understanding and documenting these, we can say something about what the potential causes of these outcomes might be. And our work addresses these types of questions in relation to mental disorders, and in particular in relation to severe mental disorders, in particular psychosis or schizophrenia. And around about 20 years ago, the generally held view was that the occurrence of psychotic disorders, the incidence of psychotic disorders in populations, was fairly constant across place and across social groups. And our work, and in fact the work of many others in the time since, has shown that this is not the case, that in fact the incidence varies quite markedly by social group and by place. So for example, in a study that we conducted a number of years ago involving around about 500 individuals with the first episode psychosis, we showed that the incidence was much higher among certain minority ethnic populations, in particular amongst the Black Caribbean and Black African populations, and other groups have shown similar findings in relation to other groups in other settings. A much more recent study that we've conducted that included around about 2,000 individuals with a first episode psychosis across 16 centres in 5 countries in Europe showed quite marked variations in the incidence of psychotic disorders, around about an 8-fold variation across different geographical areas, with the highest rates being in northern European cities such as London and Paris, and the lowest rates being in rural areas, but also in southern European cities. So in London, for example, the incidence rate is around about 60 per 100,000 per year. In rural areas and in southern European cities, it's more like 10 to 20 per 100,000 per year. And if you look at even smaller areas, what we find is that the incidence varies even at smaller levels, so that areas characterized by high levels of social fragmentation and disadvantage appear to have higher rates of psychotic disorders. And so the key point from this is that psychoses are socially patterned disorders. And what's interesting about that is it suggests that, at least in part and for some, the origins of these disorders are rooted in social conditions and social experiences. So another strand of our work has then been to try to understand what those kinds of conditions, what those kinds of experiences might be that increase risk. And one particular challenge is that if you think about stressful, difficult life experiences and unpleasant contexts, these increase risk for a whole range of disorders, a whole range of mental disorders. So the question then becomes, are there any specific types of experiences and contexts that particularly increase risk for psychotic disorders? And we've been trying to explore this type of question in a new study, a new case control study of around about 300 individuals with psychotic disorder and 300 individuals without a disorder drawn from the local population. And I want to just highlight 2 findings from this. Firstly, when we compare cases and controls in this study and look at particular types of experiences during childhood, what we find is that those experiences that are characterized by extreme, severe levels of threat and involve violence, that they increase the likelihood, they increase the odds of being a case of psychosis much more so than other types of experiences. So these are the kinds of things, for example, like growing up in a household that involves domestic violence and other forms of severe trauma. Secondly, the odds are highest amongst those exposed to multiple adversities. And what this together suggests tentatively is that what's particularly important for psychotic disorders is exposure to multiple combined adversities over time that involve, in particular, extreme threat and violence. And And I just want to finish by highlighting two new studies, two new pieces of work that we're doing, which extend this work forward. So the work that we've done so far in case control studies is cross-sectional and it involves looking backwards. We've recently established a cohort study here in South London, which is tracking the development of mental health problems across time amongst a sample of 4,000 adolescents, trying to look at what impact these kinds of social conditions and circumstances have on the development of mental health problems through adolescence. And finally, all of this work, in fact, most research on psychosis, is focused on high-income countries. And the question then is begged, do these findings extrapolate to and generalize to low and middle-income settings? And we're conducting new research in India, Nigeria, and Trinidad to assess this kind of question. Thank you.
Now again from social science to neuroscience. Dr. Anthony Vernon, senior lecturer in the Department of Neuroscience.
So thanks very much, Carmina, and thank you to the organisers. It's a great event for the opportunity to share some of our work here. So as Carmina just alluded to, we shift gears again from some really interesting social epidemiology into more sort of clinical translational neuroscience. So this is a rather busy slide, so I'll try to walk you through it as best I can. So essentially, although for many years we've known that schizophrenia is a sort of heterogeneous psychiatric disorder, which is still very much explained by its behavioural symptoms. However, in the last sort of half century, advances in genetics, epigenetics, but particularly in non-invasive brain imaging, have given us a number of interesting insights into the disorder, particularly using MRI to look at abnormal structure and function of the brains of the patients with these disorders. However, although despite these advances, it's been still very difficult to link these abnormalities that we can detect to specific etiologies, which we know are complex mixtures of genetic and environmental factors. We have other problems in terms of confounds, particularly around medication, and I'll show you an example of that. And so there's been also a difficulty here that whatever we see, we can't unambiguously attribute that to some kind of cellular molecular mechanism. So the sort of the biological abnormalities that drive these effects are still relatively unknown. So on the other side of this sort of rather great divide, you have a lot of people like myself who are working with rodent models or animal models for these disorders. Now, of course, these are uniquely human disorders, so animal models cannot be expected to recapitulate all the things you would like to see, but they can be very useful to test specific hypotheses around etiologies, particularly high penetrance genetic factors like 22q, maternal immune activation, cannabis, which we've heard about already, and also to identify confounds related to medication. So what my lab does is to try and use the animal models in a different way, which is to combine advances in preclinical imaging to now use the same level of analysis between the clinical and the preclinical domain in order to try and close this sort of translational gap. And we do this in collaboration with colleagues at the Brain Centre here at King's, which is our new 9.4 Tesla system. And this has a number of advantages. The first is that we can directly link specific hypotheses and either genetic or environmental factors to brain abnormalities. But then we can also directly relate them to their behavioural outcomes in terms of neural correlates, but more importantly, their cellular and molecular mechanisms, hence maps to mechanisms. So how does that actually work in practice? So I just want to show you two examples, if time allows. The first is this very sort of clinically relevant question, which is around the issue of antipsychotic drugs. So this is, on the one hand, the clinical evidence. So this is just maps of cortical thickness, some of which you've seen already. And here, these are patients who are medication naive with schizophrenia, and these areas in yellow are parts of the cortex that are significantly thinner, and you can see the effect size here. And these groups here, which you can appreciate look very different, a lot more red, suggesting rather a negative effect. These are patients who are typically on second-generation antipsychotics, these are people on first, and then these are people on a mixture of both. So this is in over 5,000 cases and controls, and it rather suggests that there's a negative association between the duration of medication exposure and this cortical thickness abnormality. Now, of course, you still can't discriminate as to whether that's illness progression, illness severity, or other factors that are difficult to disentangle. So this is where now the preclinical domain can answer a tractable and useful question. So here we can take a rodent model of clinically comparable antipsychotic dosing. So we use osmotic pumps to deliver drugs over chronically to the animals over time, and then we can scan them in exactly the same way as we do in humans and use the same type of analysis. So even allowing for differences in anatomy, we get the same outcome. So what happens when we do that? What you can see here are maps of cortical thickness differences against both antipsychotics and either haloperidol or olanzapine separately compared to placebo, and then against each other corrected for multiple comparisons. And what, sum a long story short, what we found is there's thinning in the frontal cortex, but also in the temporal area, which match up rather nicely with these areas of red in the human data. And so what about the mechanism? Well, now if we were to imagine drilling into this particular part of the brain, we've looked at neurons, we don't find any deficits there. We wonder about synapse loss, and Elise, who's a postdoctoral fellow in my lab, is currently working on that. But what we found is really intriguing are very large changes in both astrocytes and microglia in terms of their activation and proliferation. And this is something Marie-Caroline Cattell in my lab is now following up on. So what about the illness itself? And just in the last 30 seconds, we know that as psychosis develops, there are progressive abnormalities in cortical grey matter volume. So this is from a longitudinal study in high-risk subjects where the frontal cortex is developing at a different rate to non-converters. And we could find some associations to the immune system here. So for baseline levels of inflammatory cytokines, we're predicting this, and I've run out of time. But the take-home message is we see the same thing in the animal model, and now we can figure out the mechanism. Thank you.
Thanks. And now the last speakers of the first part of the Parade of Stars, Dr. Lucia Valmaggia, Senior Lecturer in Psychology.
Thank you, Carmine. So I'm going to tell you a little bit about virtual reality and how we use it in mental health research as well as treatment. So the key aspect of virtual reality is that it enables you to bring reality, and especially social reality, social situation, in a controlled environment. So traditionally what we do is we ask somebody to come, for example, in this case, for a neuropsychological test, and it's a very controlled experimental environment, but of course has nothing to do with the reality that these people have to face in their daily life, of course, for example, taking public transport or using, being at work or going shopping. So you could bring them, for example, here to a shopping mall and ask them how they're doing or measuring their response. But of course, you have no experimental control. So the ecological validity is high, but you have zero experimental control because the supermarket will change every second. You cannot use the same people with different participants. And what virtual reality enables you to do is to have high experimental control and at the same time, high ecological validity. When you wear a headset, you really feel like you are immersed in that world that you're seeing through the headset. You hear the sounds. If something comes at you, you move away. So it's not just what you think and how you feel, it's also how you behave, and importantly, how your emotional and physiological response is triggered. So how have we used it in the virtual reality lab? Well, we used it to look at psychological processes and mechanisms associated with the onset of psychosis, but also with the maintenance of psychosis, for example, voices or paranoia. We looked at how to use virtual reality within an assisted therapy context. So what that means is that you build it in within cognitive behavioral therapy, and then you help the person to practice with situations that they might find very difficult in real life in a virtual reality environment first. For example, here you see a pub that we developed with service users who have paranoid ideation, and we help them to overcome those first in the virtual reality before they practice in real life. We have also developed a virtual reality school canteen for the younger kids who take part in the REACH study that Professor Morgan just told you about. And we're looking there at resilient factors. So what makes you resilient to stressors are also, and we've been doing this for over 10 years, so a lot of people think virtual reality is new. It's been around for a long time. What's new is that gamers got interested, so it became more available. But so what we also did is how do you respond in real time to a medication? So if I give you a compound or I give you a placebo and I put you in a social situation, how do you respond to that? Body image and eating disorder is an area that we are venturing more and more into, looking at how you perceive your body, but also how you process clues that have to do with food. We just received some money to look at autism, especially social functioning in teenagers with autism, and how you make friends and keep friends. And with Dr. Stephanie H, we looked at unconscious bias in mental health professionals. So we are developing a training. And that brings me to the next slide, and my last slides. So virtual reality is booming. Thank you, gamers. So they made it much cheaper. Not just the equipment is much more portable, as you can see here, is quite portable and is becoming wireless. But also they made it cheaper to program. It's become healthcare, and virtual reality is a big area of investment, and also in education. I think moving forward, we have more and more opportunity to work with industry closely and to look at the clinical implementation of these strategies. But also, I get almost every week now an email from somebody across the IoPPN, King Cell Partners, and KCL at large, of people who want to collaborate and do something new with it. And very interesting, which we have not tapped into, aside from what I was mentioning about Stephanie H, is also what we can do in training. Simulation training of new mental health professionals, or indeed other health professionals, as well as, for example, helping family members understand what psychosis is, if your kid has psychosis. So there are a lot of opportunities, and I hope you are a bit more enthusiastic than you were before about what virtual reality can bring you. And, great.
Right, I think we'll begin. Thank you. Just before I move on to the next speaker, a reminder of some RIC events coming up later this year. So Operation 1 Million continues in June. In July, we have a one-off event about small grants, big vision. This is relating to applying for funding for pilot work or for early career researchers. So smaller scale grants, and we have several speakers coming from different funders that provide this kind of startup funding. So that's a really recommended event. And then in September, the proposal writing lectures will happen again, the annual lectures, and the monthly proposal writing workshops will kick off again. There'll be adverts about that in the next month or so, as there will be for Senior Thrive, which will start again this autumn. So look out for all of the emails about those coming through to you with our usual REC banner. Without further ado, we move on to Sam Heisman, who again is a much valued member of our professional services staff, and as Carmine indicated, we really like to use this opportunity to acknowledge the huge contribution that these unsung heroes have to the work that we do. So many thanks, Sam, for representing the IT community.
Cool. Thank you, and welcome back, everyone. I'm going to give a brief overview of how IT contributes to the IoPPN's success. The synopsis for this talk began with the question, what do successful research and education both have in common? Of course, this is not IoPPN specific, but their reliance on IT as an enabler to ensure success is key. Now, I'm not saying that the education and research pieces couldn't happen without IT. It's just that we make it a whole lot easier for you. I'll spend the next couple of minutes looking at some examples of the obvious and the not so obvious ways that IT provide, some of the obvious, not so obvious services that IT provide and support to make this happen. So example one, I'm stood at this lectern talking to you, and hopefully you can all hear me and see my presentation. And this is all enabled by IT's AV team. The lecture capture light is on today. And this is one of the most popular IT services, especially when it comes to exam revision time. Behind me, now tucked out of sight, is a wireless access point run by IT, which means that if any of you are currently connected to Eduroam or the guest wireless service, the cloud, you're using services provided and supported by IT too. And it's great that if you go to any of our King's Health partner sites, you get the same services there. I love the fact that here at Denmark Hill, you can get internet access via the wireless services from both the Maudsley and King's hospitals.
This took a lot of work to implement, as the hospitals are fundamentally about keeping data secure, whilst as a university we're about information sharing. It's a great example of how we can work together to utilise technology to make people more efficient. If you use the student computing rooms, a computer in the library, a lone laptop or anything with the standard operating environment on it, you're using IT services. Let's think about the applications you're using on them. What goes on behind the scenes to get them there? Well, we have someone responsible for negotiating the purchase of those applications and ongoing licensing. We have a team responsible for packaging them, ready for deployment. And this includes testing them to make sure they work, because there's no point deploying applications which don't work. And once they're deployed, they need to be maintained, and we do all of that too. We work with the faculty to make sure that they're the right applications to be delivered, and if no one is using them, what's the point in having them? And we can track usage and provide the data into helping make those kind of decisions. Want to print out a piece of work? That's IT too. Want to save your work somewhere? We provide the storage, file storage, including backups. And we have a security team working hard in the background to ensure that the university's IT networks and systems are secure and GDPR compliant. In our current cyber risk world, there is constant monitoring and adjustments being made to keep our data safe. Getting the basics right, of which many of these examples are, has been the focus for IT over the past few years. Now we have resilient and consistent foundations in place. We're in a far better position to support the research environment and address some of the challenges you face. Two of the research-specific services we provide include the high-performance computing cluster, including Rosalind, and the very important King's Research Portal, Pure. Now, I've got to give out a big shout out to the clinical trials unit and the mouse management people as well, because we helped with that. And this is a really exciting time for research IT, and significant service improvements are imminent. More collaborative examples include all the telephony services. We're changing technologies which mean that you no longer have to be at your desk to use a phone. Skype for Business allows you to take your number with you. And Skype is just one of our collaboration tools. And for those who aren't familiar with it, I do recommend trying it out. It's a great way to message, talk, and share what you're working on with people wherever they are in the world. If your mobile phone has run out of battery, you can use one of the charge boxes to recharge it, and you'll find them dotted around the campuses. And one of the first interactions people have with King's is our website. And whilst IT run it, it enables you to get your messages across to your various audiences. And with the new version, it's much easier to reach them on a wider range of devices. If you've needed IT support, you may have visited one of our tech bars for immediate face-to-face assistance, or you may have called our service desk, who are based in a rather splendid tin shed in Cornwall. Lavington Street will shortly become the new home for the rest of IT. Remedy Force is our lifeblood of dealing with your requests, and that's why we always ask you what your ticket number is. So, how do we deliver all of this? IT is organised into three main areas. Solutions, who are responsible for designing, building and integrating new services or delivering major upgrades to existing ones such as SITs. Services, who run over 300 services, including the ones I've mentioned already. And our Office of the CIO, who help ensure things go right. Of course, IT is never going to be perfect. The Armageddon episode a couple of years ago is testament to that, and we have to acknowledge, understand and learn lessons from the past. Over the past few years, IT has been on its own journey. It's grown in size, scale and maturity, and you can find out more information about this, our teams and roles, and where we're going in future to ensure your continued success on the IT web pages, or by contacting me or the service desk for more information.
Thank you very much. Next we have Dr. Jonathan Coleman speaking about genetic influences on depression.
Hello. So, as Talia said, my name is Jonathan Coleman. I'm a postdoc at the SGDP Centre, and I primarily work looking at how genetic influences, so changes in the DNA code across the genome, interact and influence psychiatric disorders and also the experiences that people who have psychiatric illnesses have in their daily lives. Most of my work recently has been focused on one particular sample, the UK Biobank, and this is a fantastic resource of 500,000 individuals who were recruited in the last 10 or 15 years, and they're aged from 40 through 70, and it's a study of the ongoing health and ultimately late life health experiences of the country as a whole. More recently, we've had a targeted psychiatric health questionnaire in the UK Biobank, and this has enabled us to look at a variety of different psychiatric outcomes. And the one I'm going to focus on today is the interaction between traumatic experiences, so that is the T+, you'll see there, and meeting depressive criteria, which is the D+, you see there. And as you can see from this slide, we have around 10,000 individuals in each of our groups there, and around 40,000 people who, while we have around 40,000 controls, we also have a good spread of people who both receive, who both experience traumatic events and are depressed, and who don't experience traumatic events and also meet depressive criteria. So what we're particularly interested in is how can genetics inform us about these two things, and do we see a difference in the genetic basis of depression in individuals who are exposed to trauma and individuals who are not? So this is a fairly wide-ranging piece of work, and I'll focus on two findings that I think are particularly interesting from this work. So the first is this, which is the genome-wide association study of trauma exposure. So in this, we look at millions of variants across the whole genome, and we compare them to see whether that variant is more enriched in one group compared to another. So in this particular example, we're looking at individuals who report trauma exposure. And going across the graph, so at the bottom we have the position of each variant, which is each of these crosses on the graph, moving from left to right across the different chromosomes and across the different positions on those chromosomes. And then on the y-axis, we have the minus log 10 p-value of the test of whether the gene is enriched, or the variant is enriched in one group over the other. And what we see, as you can see from the graph, is 6 regions that are associated with traumatic exposure. And what this means is that we can take the genome, we can take someone's genome, and we can learn things about their environmental experiences directly from their genome. What we now need to do is understand how those genetic correlations affect real life and why those genetic correlations are associated with traumatic experiences. What is potentially of interest within these particular loci is, for example, FOXP2 is a locus that has previously been associated with a variety of psychiatric outcomes, including language development and ADHD. And that's true of several of the other loci we see as well. So one potentially interesting concept is the idea that the people who we get who are reporting traumatic experiences are also showing behavioral or psychiatric differences, or potentially the people around them who are related to them are also showing behavioral and psychiatric differences. So there are many potential reasons why we have this relationship between the genome and the environment. I also said I would talk a bit about depression, and so here we have, here rather than looking at individual genomic loci, we're looking at the gross genetic influences on the trait. So when we look at the gross genetic influences on depression, we can compare them to the gross genetic influences on other traits, such as in this example, body mass index or schizophrenia. We can find similarities and correlations between depression and that external trait. Specifically, what we see here is that while we see high genetic correlations between depression and other psychiatric disorders, which is something you see for all psychiatric disorders, those don't really differ between people who are exposed to trauma and people who are not. However, when you look at the top one, body mass index, which has a positive correlation with depression, this seems to be much stronger in individuals who are trauma exposed than those who are not. Finally, we also see a slightly strange association that we haven't really fully understood yet, which is that we don't see any association with femoral bone mineral density, so the density of this bone just at the top of your leg. In fact, that one. We see no association with that in depression or in people who are depressed and exposed to trauma, but we see a strong negative genetic association between the two in individuals who are not exposed to trauma. So we're seeing something that maybe is cryptically hidden because we're not being able to split groups of depressed people into trauma-exposed and trauma-unexposed. So thank you very much, and there's a preprint available on bioRxiv if you'd like to read it.
So next we have Dr. Faith Matcham, who's from the Department of Psychological Medicine.
Thank you very much for inviting me to speak today. I'm going to talk about a project we're working on at the moment called Remote Assessment of Disease and Relapse in Major Depressive Disorder. Outcome measurement in chronic illnesses are usually measured through research designs where patients come in at pre-designed moments in time to have their outcomes measured, which don't necessarily reflect the fluctuations they might experience in symptoms over time. Alternatively, we can use data collected as part of clinical services, which in turn are likely to show symptoms reflective of people when they're experiencing a crisis, heightened severity of symptoms, particularly in mental health services, which will have triggered that particular appointment. What we're missing out on at the moment is everything that happens in between that comes before that moment in time when we have a patient or a participant in front of us. And what we're hoping to do as part of this study is find new ways of measuring the lived experience of chronic illnesses and long-term conditions, which can give us insight into how people are feeling over time. So to do this, we're using remote measurement technologies. This can include wearable devices. Depending on the make and model of the device, they can measure a whole range of aspects relating to daily life. Behaviors like physical activity, sleep patterns, actigraphy can tell us people's movements, the direction that people are walking in, how far they are away from the Earth's core. We can measure heart rate and stress and emotional reactivity through heart rate variability, electrodermal activity, body temperature. There's even some devices out there which allow you to measure respiration, so the extent to which people are breathing in and out, so you can see whether they're participating in more intense physical activities. Other types of remote measurement technologies include the sensors that are found in most modern smartphones. This can tell us a whole load of information about how people are living their lives. GPS can tell us about people's location, whether they're leaving the house, how far they're going, patterns and daily routines that may be indicative of a healthy or a dysfunctional behavior. Sensors like Bluetooth connections, Wi-Fi hotspot connections, payment data can even tell us how people are interacting with other aspects of technology in their daily lives. We can also gather information from call logs, message logs, how people are interacting with apps, internet usage, to tell us about social activities, people's social environment, and how they're interacting with people they know. There are other sensors, such as microphone sensors, that can give us an insight into the amount of ambient noise in someone's environment or the amount of light that is in their environment. And we can even measure how people interact with their phone screen, and there's some evidence to suggest that how people interact with the phone screen can be indicative of cognitive state. Smartphones also offer an opportunity to develop apps for symptom measurement and personal experiences, so we can combine what we're calling this passive data collection, which is unobtrusive, doesn't require any real input from participants. We can combine that with data collected via standardized questionnaires, which can be delivered via a smartphone app. So I work as part of a large project called the Remote Assessment of Disease and Relapse in the Central Nervous System. It's an international consortium which is aiming to look at the use of these technologies in a range of conditions, including multiple sclerosis, epilepsy, and major depressive disorder. I'm primarily involved in the work on major depressive disorder. We are running a prospective observational cohort study, a non-interventional study, which is aiming to see if using this kind of technology is acceptable to people, whether it's feasible and provides useful data that is useful in any way to tell us about people's clinical state, or useful for predicting future deterioration or relapse. It's very early days in the study. We only started recruitment in October, but I wanted to share a little bit of data we have from two of our participants. This is GPS data from two participants. On the left, you can see this individual. They've participated for about 90 days. They've traveled about 12 kilometers from home in total, is the longest they've been from home since they started participating, but they make lots of frequent trips. The participant on the right, again, stays very close to home, but has traveled nearly 1,200 kilometers from home twice during the period of participation so far. So this gives us a little snapshot into people's daily routines, and we might be able to identify personalized predictors of relapse and early indicators that mental health may be declining. Thank you very much.
So next we have Dr. Ben Gardner from the Department of Psychology telling us about psychology of the workplace.
Thank you. So I'm a social psychologist by background, but I spent the last 10 years working in health psychology, and I'm going to be talking you through some work that I'm currently doing, which kind of sits in the interface between social psychology and health psychology. I'm currently interested in sitting time, and the reason for this is that sitting is emerging as a new health behaviour, by which I mean that there's evidence emerging that the time that we spend sitting for us is bad for our health. Sitting time has been linked to premature death, to some cancers, to diabetes, and it's also been linked to adverse mental health. And what's more, it's suggested that the time we spend sitting is bad for us independently of physical activity, which suggests that it may be the case that perhaps we can't run off our sitting time. We can't compensate for our sitting time by being more physically active. So the thing that we need to do to improve our health is not only be more physically active, but also spend less time sitting. Now, if sitting time is a problem, then office workers are at particular health risk because our earlier research has shown that office workers tend to spend 2/3 of their working day sitting down. And over the course of a waking day, the average office worker here in London spends 10.5 hours sitting. Now, given that it's estimated that half of the UK workforce is based in offices, then this is a real public health problem that we've got on our hands. So what's been done? Well, interventions have been developed. Researchers have gone straight to the intervention stage to try and reduce the time that people spend sitting. But my concern with this work is that people haven't really tried to understand the psychology of sitting, why people sit, how acceptable sitting reduction is. And as a result, we seem to have interventions at the moment that are quite unrealistic. They typically involve an office worker being taken out of work for around an hour on multiple occasions and having detailed behavioural counselling sessions with someone about how to reduce their sitting time. If you ask me, employers would not buy that kind of intervention package. It's fundamentally unrealistic. What's more, it's not informed by an understanding of why people sit for such long periods. So what we're currently doing is a program of work to try and understand the psychology of workplace sitting. And all of this is leading us towards the development of an intervention that should be realistic and hopefully implementable and feasible for implementation in the modern workplace. So I'm just going to talk you through a couple of studies that we've done recently as part of an MRC-funded grant. The first of these was quite a fortuitous one. Just as our grant was starting, we heard of this breaking news story that an expert consensus statement had been published suggesting, well, it's the first statement that suggested how long people should stand for in the workplace, and it kind of put a figure on the amount of time people should sit and the amount of time people should stand, and it was really quite notable, and it attracted a great deal of media attention. Now, what we realised was that the media attention it received, there were stories about this published on newspaper websites, and several of these websites had sections where readers could comment on the story. And we realised that these comments gave us a really nice indication of public acceptability of the sitting reduction message. So here is, I think this was how it was reported in the Daily Mail. Then you get people writing stuff like this, that this is a movement to try and return this country to Victorian work practice, and so on and so forth. But as I said, what we were aiming to do was synthesize these. We found that there were 5 sites that had reported this story, and across these 5, there were 573 unique comments. Most of them from the Daily Mail, but that's another story. Anyway, so we applied thematic analysis procedures to try and find out what was going on here, and what we found that there were 3 types of response, essentially. One response was where people challenged the credibility of the statement that was put in front of them. People saying, I don't think this is novel, I don't think this is telling us anything new. Some people questioning the authority of the authors. One person saying, well, this comes from a university where there isn't a medical department, so these authors can't be doctors. Of course, complete nonsense. Then some other people also challenging the credibility of public health as a discipline, saying that public health is actually all about scaremongering. We shouldn't believe these people, they're all out to sell us things. And lastly, we found we found evidence of a more kind of receptive response, which saw the statement as a spur to action. So these were people who were accepting of the message and offered strategies, tips and advice for other people on what they could do about it. Very quickly, we followed this up with a study of standing in meetings. Now, it's proposed that one way of getting people who work in offices to reduce their sitting time is by getting them to stand more. So what we did was we had people stand in meetings and we interviewed them about their experiences. What we found was that people typically found that standing physically and psychologically removed them from the group, and so to avoid this, they put themselves in the corner or the side of the room, and also that they felt awkward, they felt like they were being attention seekers, and so to avoid this, they positioned themselves in certain places, and they felt that standing was not appropriate in particular meetings. And lastly, some people felt that they had to give an excuse to other people as to why they were standing, and they felt that some excuses weren't good enough, and so they made up that they had a health reason that meant that they had to stand. As I said, all of this is now leading us towards the development of an intervention to reduce workplace sitting time. Thank you.
Our next speaker is Dr. Nikolai Vysokov. Did I say that right?
Yes, thank you very much.
From Wilson Card.
This is going to be a jump from the previous talk. I'm going to talk about cellular and molecular biology. So the next 5 minutes I'm going to talk about pain. So a painful stimulus, when it excites the nerve endings, the signal travels through into the spinal cord it's transmitted by the dorsal ganglion neurons into the spinal cord, and then there it's relayed up into the brain, and it's in the brain that we feel this painful sensation. Now this is actually a very advantageous evolutionary mechanism to prevent from being burned or severely injured, but in neuropathic pain, this mechanism is malfunctioning. People are feeling pain in the absence of any painful stimuli. So the problem with that is that there is no adequate, specific, and effective treatment for this type of pain. And it isn't there because studying injury-induced pain in animals, particularly in humans or rodents, is a serious ethical issue. So our solution to this is a cellular model of pain, recapitulating aspects of this peripheral nerve circuitry using a 3-compartmented device. So just to explain here, in the middle compartment, we place these dorsal ganglion neurons. They spread their axons in both directions. So on the one side, we've got them synapsing with neurons from the spinal cord dorsal horn. And on the other side, there is nothing to synapse with, so they act as a model of the free nerve endings. So this is what it looks like in reality. If we put a red dye in this periphery compartment, this dye gets taken up by the dorsal ganglion neurons, it gets transported into the middle compartment, and then what you see here in red, where the red arrows are pointing, in the spinal cord compartment, you can see those axons labeled with red. This dye can only come from DRG neurons which have spread their axons both ways at the same time. Moreover, what we can do with this is we can add a genetic indicator of nerve cell activity. So here on this picture, we can see on the left is the DRG axons in the spinal cord compartment being activated. So when you stimulate the periphery, we can see that the DRG axons get activated in the spinal cord compartment, so the signal travels all the way through. And moreover, in this spinal cord compartment, what we see is when we use another dye for looking at the activity of the spinal cord neurons, we see that that signal gets transmitted across the synapse from one cell type to the other. And we can look even deeper into the mechanisms of this transmission of the signal by using a technique called patch clamp electrophysiology, where here you can see that each peak corresponds to a complex synaptic event. So what about injury and neuropathic pain? Well, one of the really cool things that you can do with this device is you can apply shear pressure to the periphery and cut off the axons. Then what we do is we put this dish back into the incubator for another 3 days, take it out, and then start looking at the nerve cell activity. And as you can see here on the right-hand side, on the bottom right, in the injured cultures, we see that the dorsal horn neurons, or the spinal cord neurons, are being spontaneously active in injured cultures, but not in control. And we can also see this as, when we look at the electrophysiology of the signal transmission. One of the interesting things that happens with it is that in some cells, this spontaneous activity comes in waves, which are representative of some of the reports that people with sciatica or peripheral nerve injury have found, this sort of, activity of 2 or 3 times per minute, this kind of ongoing pain. So, with this model in mind, because the cells have been sitting in the incubator for 3 days, they observed spontaneous activity and a change in pharmacological profile of the cells, representing what happens in humans, what we observed in clinical trials. This cellular model allows us to research chronic effects of injury-induced neuropathic pain in the dish. Thank you for your attention.
So our next speaker is Stephanie Hines, who's another one of the finalists from the PhD 3-minute thesis competition. So congratulations on that as well.
Okay, so imagine you're walking down the street and suddenly a lion jumps out at you. Your heart starts pounding, your breathing gets quicker, and more blood is pumped to your muscles, readying you to run. This is a fight or flight response, and in this situation, it's life-saving. Now imagine feeling like this when leaving the house, remembering a past event, or even chronically. This happens in conditions like anxiety and post-traumatic stress disorder. At this point, it becomes debilitating. The opposite happens in other conditions, like familial dysautonomia, where the system that elicits a fight or flight response degenerates. This causes dysregulation of the individual's organs and is eventually fatal. We know that the fight or flight response causes the release of adrenaline into the body, but exactly why these exaggerated responses happen in some people is not entirely understood. Potentially, it could be traced back to the development of the sympathetic nervous system. The sympathetic nervous system is a network of nerve cells that send signals to your organs and muscles and is under the involuntary control of the brain. It has 2 main roles. One of maintaining the body's internal balance state and the other of eliciting the fight or flight response. When there is perceived or physical threat, it changes the body from being in this internal balance state into one ready for action. An important part of it are the synaptic connections between spinal nerve cells called preganglionic sympathetic neurons and an organ called the adrenal medulla. The adrenal medulla is what releases adrenaline into the blood and is mainly comprised of a cell type known as chromaffin cells. This release of adrenaline causes mobilization of the body's resources and redirects them to the muscles, preparing the body for action. The aim of my PhD is to understand more about how these neurons and chromaffin cells develop and form these synaptic connections. To do this, I'm using stem cells, which are cells that have the ability to become any other type of cell given the correct instructions. However, we don't yet know all the instructions needed to make these types of cells, so that's where I've started. So far, I've turned stem cells from mice into a mixture of different types of spinal nerve cells. I confirm this by seeing which genes they express. One of the genes they express is HB9, which here is labelled in green. This shows that they are a subset of spinal nerve cells that are still developing. This is important as I want to take cells from this subset whilst they're still developing for the next stage of my experiments. I'll see how they form these synaptic connections. So next, I want to confirm that some of this subset are in fact preganglionic sympathetic neurons. To be able to do this, I'm currently exploring which genes are expressed in these cell types in the developing mouse embryos at different time points to find a specific marker for these cells. And I can confirm I have made the preganglionic sympathetic neurons. I will then alter the stem cell's environment whilst they're growing to try generating more of them. Then I'll perform a similar set of experiments for chromaffin cells. When I have the 2 cell types, I will combine them into compartmentalised devices which are made by our lab. These have two different compartments where I'll put in the two different types of cells, but they have channels connecting them, allowing the neurons to send nerve fibres through and connect with the chromaffin cells. This mimics their natural environment, where nerve cells have to send their projections over long distances to be able to connect with their target cells. Using this, I'll be able to see more about how these connections are made and behave. I'll use techniques like optogenetics, which involve genetically altering the nerve cells so they express a light-sensitive protein. When you flash a blue LED light at these nerve cells with this protein, it causes them to fire, allowing me to see if these connections that are being formed are functional and if they are in fact releasing adrenaline into the cell environment. Then, I hope to develop a clinically relevant model using human stem cells. These can be made from patient tissue, so I could potentially see if there is a genetic alteration in these patients with a debilitating fight or flight response that causes a difference in these two cells and how they interact and behave. This is something that other ways of modelling these diseases are not able to do. Potentially, we could learn more about conditions like anxiety, post-traumatic stress disorder, and familial dysautonomia, and this could lead to potentially more effective treatments in the future. Thank you.
So our next speaker is Dr. Sylvain de Rivières from the SGDP Centre.
So why do some of us behave differently than others? And why do some go further and develop conditions that are associated with neurological and psychiatric disorders? We know that this is partly due to variations at the level of DNA. We have, for example, genetic variation that we inherit mostly from our parents. There are also epigenetic variations that we acquire throughout our lives, either due to normal development or because of our exposure to different experiences throughout life. And one of these epigenetic variations is DNA methylation. We also know that neuroimaging measures, as we've shown earlier this afternoon, are good correlates of health-related outcomes. What we know less are specifically which genes or genetic variants do contribute or affect brain structure and function, and thereby create a risk for disease. So we are using a combination of genetic analysis and neuroimaging analysis to study this, and I will present some of our key findings over the years. I have to acknowledge that this work is due to our contribution to several international neuroimaging genetic consortia, including the Imaging Consortia that is in Europe, CIVIDA in India, and Enigma worldwide consortium. So what have we found over the years? So concerning our genetic analysis, so we have found that it is indeed possible to identify gene variants that affect brain structure and function and thereby contribute to disease risk. So this is exemplified in the example that I highlighted here that resulted from a large genome-wide association studies of subcortical brain volumes in over 30,000 individuals. And these study identify several genetic variants that really affect specific volumes, subcortical volumes. As I have highlighted here, there are variants that associate with, specifically with the volume of the putamen. So we have conducted functional analysis to show that those variants that associate with the volume of putamen contribute to risk of to ADHD symptoms in adolescents and affect neural correlates of ADHD, but they do so in a sex-specific manner. What we also have learned from our genome-wide epigenetic studies is that we can collect very useful information from blood DNA methylation patterns, and this information informs us about brain structure and function. So we first showed that in a subsample of the IMAGINE study in about 500 individuals. So what we found that DNA methylation in a specific gene, PPM1G, correlate with brain structure and behaviors that are predictive of future psychopathology, and is, in this case, predictive of alcohol drinking behavior and future alcohol abuse. So this study was really important to us because it showed that actually we needed larger studies of this kind to analyze the link between peripheral DNA methylation patterns and brain structure and function. With this in mind, we set up the Enigma epigenetic working group that I lead, and in our first epigenome-wide meta-analysis study of brain volumes that we're about to publish, we identified DNA methylation patterns that really associate with brain volume consistently in the 11 cohorts that we analyzed so far. And the good news for us is that those DNA methylation patterns are annotated to genes that are really important, relevant for hippocampal function. So really showing that blood DNA methylation profiling hold promise for identifying biomarkers related to brain function. So what's next? So we're trying to expand our analysis and using big data approaches to identify early biomarkers of disease. And in a specific project I highlighted here, we focus on eating disorders. So we do that in analyzing the large data set and the data collected in IMAGINE, and also recruited new patients with eating disorders to validate those biomarkers discovered. I thank you for your attention.
Our next speaker is Dr. Kinga Baskanyi. Close enough?
No, very, yeah.
And she's from the Center for Developmental Neurobiology.
Okay, hello everyone. First of all, I would like to thank the organizers for giving me the opportunity to speak. And I'm going to tell you how neurons in your brain decide whether to be or not to be. And in our case, that's an actual real question. So they really do decide whether they should live or die. Yes, and I need to make a note of this, but I will come back to this at the end because I assume in 5 minutes' time everyone's going to forget this. So, how does our brain actually work? Now, I'm a fundamental scientist, but I fancy having a bit of a scope, so I'm simplifying it and bringing it to a new level at the same time. It's a bit confusing, but hopefully you will get to follow me. So I would like you all to imagine our brain cells themselves taking part in a dinner table conversation, okay? Chatting away, somebody talking, others listening, some deciding not to listen, clearly forming an opinion about what's being said. What would happen if everyone decided to talk at the very same time? Not very much would come out of that discussion in terms of conclusions, right? Or what would happen if everyone fell quiet at the same time? That's sort of what we are all dreading, unless we are talking about a talk given by me and listened to by you. But in our case, we are talking about a dinner table conversation. So neither of these scenarios would work. So what we are doing in our lab is that we are taking quite young 5-day-old mouse pups, and we put in an imaging window on the top of their head, which you should literally imagine as a window. It's a piece of glass that we can implant in, and then we can put them under the microscope and actually come back day after day and look at the exact same cells. But what we noticed is if you pay attention to cell number 32, it has disappeared from one day to the next. So it's become quite quickly a murder mystery, really. Where did that cell go and why did it go away? We cannot just only simply look at them, we can also follow their dinner table conversation through a calcium dye. So we can literally watch them talk. And if we have a look at them on day 7, which is the day before number 32 was murdered, you can see that it didn't have much to contribute to the conversation, right? So the number of the lines is reflective of the number of times they talk, and the length of the lines is how long they talk for. So compared to everyone else around the dinner table, number 32 hasn't done much, so it's gone away. So what happened to number 32? Actually, there's two different types of cells in a dinner table conversation when we're talking about the brain. The green guys are the ones that tell others to talk, so they would be the ones who would ask maybe shyer members, what is your opinion? And then we have the purple ones, my favorite ones, who shush others. They literally tell others to quieten down so we can listen to everybody else who may have something meaningful to say. When we looked at the actual cell number in the developing brain of the mouse, what we found is that, first of all, the green cells die between 2 days of age and 5 days of age. And then they are followed directly by the purple cells. So instead of a murder mystery, it's not the case of the green ones killing the purple ones. It's the case of the purple ones deciding to commit suicide because they've lost their partners from the dinner table conversation. Looks pretty dark, right? But then the idea is that at the end of the day, at 21 days old, which we declare an adolescent mouse, we have the right ratio between the ones that ask others for their opinion and the ones that tells others to shut up. And then we end up with a dinner table conversation where we can have a meaningful discussion and make a decision on a certain thing, or decide to learn something, or memorize things. So, dinner for one, not an option in the brain for a cell. But how does the neuron actually read this, and how can it decide to commit suicide? So it will need a murder weapon, its own murder weapon, and in our case it happened to be a switch that is literally switching between life and death, and this is a gene called p10. And in the mouse brain, if we take p10 away, we end up with actually 30% more of the purple ones, the shush neurons, than what we should be having. So we will have a much quieter brain altogether. So we shifted the balance instead of the everybody shouting, which would be equally bad, let's agree. So that's kind of an analogy for epilepsy. You don't get a lot of good out of that. But we actually ended up with a brain that's extremely quiet in certain situations. And in our case, this p10 gene is actually a gene that's well known to cause autism in humans. So finally, there's nothing else left for me to say than to actually remind you or tell you that next week we have our paper coming out, so please Google it. And thank the funders, which is the MRC and the Wellcome Trust. And also to remind you, please shush about this until next week, because we have an embargo and nature is quite powerful about that. So the open questions that are still remaining is, what about other areas of the brain, like the putamen we just heard, very important for ADHD? What about evolution? So is this actually playing a role in expanding certain brain areas over other areas? Do we actually have this phenomenon in humans? We don't quite know. We think we do, and if we do, then does it actually contribute to other neurodevelopmental disorders, just like autism? So with that, I would like to thank you and pass on to the next person.
So our next speaker is Dr. Alexis Cullen, who's at the Department of Psychosis Studies on a Henry Wellcome postdoctoral fellowship.
Thank you, Talia, and thank you for inviting me to be here today. So, my work is really rooted in the idea that schizophrenia is a neurodevelopmental disorder. And what I mean by that is that most of the signs and symptoms are actually present in an attenuated form prior to the onset of illness. So the shaded region here is what we refer to as the prodromal period of psychosis. So this is when individuals are unwell, but their symptoms are not quite severe or frequent enough to warrant a diagnosis of psychosis. But even before that point, we know that children who later go on to develop schizophrenia present things like psychotic-like experiences that Dr. Helen Fisher was talking about earlier, and other antecedents of illness. So this has led to the idea that we might be able to use a clinical staging model similar to what's used in other medical disciplines, for example, something like cancer, as this would allow us to track the progression of the disease in individuals and hopefully being able to allow us to give more appropriately targeted interventions. So there's a number of biomarkers that we could use to help us with clinical staging, but what I'm particularly interested in is the hypothalamic pituitary adrenal axis and the inflammatory system. And that's because those systems could really tell us about things like psychosocial stress, like disparity in socioeconomic conditions can actually get underneath the skin and give rise to psychosis. So my fellowship, which is funded by the Wellcome Trust, is really looking at whether we can use HP axis and inflammatory markers to help us with clinical staging. If we could, I think what we might expect is that these markers can distinguish between individuals at different stages. They might be able to predict who actually goes on to progress to the next stage of illness, and also they might be correlated with things that also show progression, so things like cognitive deficits. So my study takes lots of different cohort studies and assesses people at different levels across the proposed psychosis spectrum. So first up, we've got the Child Health and Development Study, which I've been involved with for the past decade. So we recruited children at age 9 to 12 years who are at elevated risk of schizophrenia because they either had a family history of psychosis or antecedents of illness. So that's telling us about the early stages of the disorder. And we're now following those individuals up at age 17 to 22 when they're all now a lot more taller than I am. Next, we've got data from some very large international studies, so SciScan running in Europe and Naples, which is running in the US. And this tells us about people who are at clinical high risk for psychosis. And by that, we mean that we think that they're in the prodromal phase of illness. And it also gives us information on first episode psychosis patients. Then we've got the GAP study, which has been going on here for some time. And this study initially recruited individuals who were in their first episode, and now we're following them up over time 5 years later to see who's gone on to stage 3, so recurrent psychosis, and stage 4, chronic schizophrenia. So my study is really attempting to collect the same measures across all of these different cohorts. So we've got self-reported measures of stress, we've got measures of HPA axis function, so that's predominantly cortisol, so salivary cortisol and hair cortisol, and also blood and buccal cells for inflammatory markers. Now I just want to share some very preliminary findings from the study and just acknowledge that most of this has come from the MSc students and the BSc students who've been working very hard on this study for the past 3 years. So first up, we've looked at a subset of the hair cortisol data, and we've got it for around 100 participants now. And you can see that in stage 1B, which is the prodromal phase of psychosis, hair cortisol is slightly elevated relative to controls. But in established psychosis, and here I've had to pull together individuals at stages 2, 3, and 4, you can see that it's significantly higher compared to controls. Next, we've shown that salivary cortisol in the CHADS cohort at age 11 to 14 years actually predicts the development of prodromal symptoms at age 17 to 21, even after you adjust for some of those other measures of psychopathology. So this is the cortisol awakening response, which is typically blunted in people with psychosis, and you can see that the more blunted it is, the higher the levels of negative prodromal symptoms and disorganized prodromal symptoms. And then finally, I've been doing some work looking at correlations with cognitive function. So salivary CRP, which is a marker of inflammation, higher levels of that are associated with poorer performance on measures of inhibition and inhibition switching when we look at children in the CHADS cohort who are in stage 0 and stage 1A. And then using data from the Naples cohort, we're focusing on, again, youth who are at stage 1B, so the prodromal phase, and here salivary cortisol is associated with poorer executive function. So that's just a little snapshot of the work that we've been doing so far, and I hope that convinces you that stress and HPA axis markers might be useful for clinical staging. I just want to thank the team and also the Wellcome Trust for supporting this work. Thank you.
So next we have Dr. Louise Hull, who's from the Department of Health Services and Population Research.
Hello, yes, my name's Louise Hull. I am going to present some of my fellowship work. I'm part of the Centre for Implementation Science, which is one of the only centres for implementation science within the country. I'm also a King's Improvement Science Fellow, and the work that I'm going to be presenting today forms part of my fellowship work. So I'm going to be talking about the development and trying to squeeze in a little bit of information about the evaluation of a tool that I've developed to support applied health researchers in embedding implementation science into their work. It's called the IMPRESS tool. So implementation science is a relatively novel discipline within the field of healthcare. So I thought it would be really important to just explain and provide a bit of context about what implementation science is. So implementation science has been defined as the scientific study of methods to promote the systematic uptake of clinical research findings and other evidence-based practices into routine practice. And the overarching aim of implementation science is to improve the quality and effectiveness of healthcare services. So there's increasing recognition among the research community that we are extremely good at developing effective evidence-based interventions, but these interventions typically fail to be translated into clinical practice. So there are a number of systematic reviews that have kind of looked at the scope of this problem, and there's estimates that over 80% of research fails to ever be translated into practice. And though that 20% that does make that leap into clinical practice, it takes on average 17 years. So, as I said, although implementation science is relatively novel, it's really key to closing this so-called evidence to practice gap. And the World Health Organization has described the implementation of evidence-based practices as one of the greatest challenges facing the global health community, and has specifically identified implementation science as key to closing this gap. So, as I said, implementation science is relatively new to applied health researchers. And so this leads on to problems in terms of actually designing high-quality implementation research. It's actually quite a complex and daunting task for people that haven't received specialist training or don't have access to implementation scientists to actually support the development of high-quality research. So to overcome this problem, I have developed the Implementation Science Research Development Tool, and this really provides researchers with a systematic step-by-step approach of incorporating the core principles and methods of implementation science into their research. It's evidence-informed, so it's based on a scoping review of the literature, and it's expert-derived. As I said, I'm part of the Centre for Implementation Science, and we're very fortunate to have a scientific advisory panel which consists of national and international experts within the field of implementation science. IMPRESS contains 10 domains that cover the core principles that researchers should consider when designing implementation research. I'll give you some examples of how implementation research is distinct from more traditional kind of effectiveness research that applied health researchers are familiar with conducting. So applied health researchers, they're very familiar with what we term service and patient outcomes, but what implementation research is interested in is a whole set of different implementation outcomes, which were alluded to in the talk about the sitting interventions. So things such as the acceptability of an intervention. Is it actually feasible to implement in practice? Is it appropriate? When people do intend to adopt an intervention, is it actually implemented as it was intended? Also, things such as unintended consequences as well. So as applied health researchers, we develop interventions with best intentions. However, what we typically fail to look at is the potential unintended consequences. Again, going back to the previous talk on the sitting intervention. So obviously one of the outcomes wasn't to make people feel awkward, but when people did implement it in practice, this was one of the unintended outcomes. Also, Impress as a tool has a supplementary guide. So this will direct researchers to relevant literature. It also has other online resources. I'm being told that my time is up, so I'm going to switch to my last slide. So in terms of focusing on dissemination and impact, I just wanted to make the point that in addition to going down a more traditional academic route, publishing in academic journals and presenting at conferences, the tool and guide is available on our King's Improvement website. It's free to download. And it's also been involved in supporting grant applications, both internally and externally. Thank you, and I'm sorry I overrun.
Thank you very much. So next we have Professor Oliver Howes from the Department of Psychosis Studies.
So thank you very much. I guess many of you will be wondering what Groundhog Days has to do with treating psychosis. Well, in Groundhog Days, the Bill Murray character is trapped in a cycle where every event each day repeats the events that have happened the previous day and the previous day and the previous day. And that's often how I feel when I'm trying to treat patients with psychosis, where we've got over 30 different drugs, but they all work in exactly the same way, and that is by blocking D2 receptors. So it just seems like you are in an episode of Groundhog Days, repeating the same thing day in, day out. So the big question that we've been struggling with is how can we go beyond dopamine D2 blockers and develop new treatments for psychosis? And actually, Groundhog Days gives us some hints there, because to break out of this cycle, Bill Murray realizes that he has to firstly do something different, secondly, he has to learn something new, and finally, he has to persevere. So we thought we need to do something different, and we wanted to look at a common substance of abuse that it reliably induces psychosis, and that's special K. So I guess some of you might have had that for breakfast. Don't worry. On the streets, they're actually referring to ketamine. And many of you all know that our work is usually using imaging in healthy volunteers and patients, but we actually wanted to develop a translational model that might allow us to test new treatments. But we use ketamine as this model because when you study people that take special K regularly, and I'm talking about ketamine now, not the breakfast cereal, they develop psychotic symptoms that last even after they've stopped taking ketamine. So when the ketamine's out of their system. So that tells us that it's inducing some lasting changes in the brain. And we went on to learn something new, which is to develop a PET measure in mice that we could use to look at the same systems that we see are altered in patients to see if ketamine is affecting these circuits. So we looked in mice who were given chronic ketamine and looked at the dopamine system and found that ketamine induces long-lasting changes in dopamine synthesis capacity, which is really interesting because this is exactly the same change that we see in patients with schizophrenia and as Samir Jauhar has just shown, also in other psychotic disorders. So here you see the elevation in dopamine synthesis capacity in patients that's related to their psychotic symptoms. So we can induce that in mice with ketamine. But that doesn't tell us about the circuit that might be involved. So the next question is, does this require dopamine midbrain neuron firing? And we are able to manipulate this using chemogenetics, which allows us to switch off the dopamine neurons selectively in the midbrain. And here you see that if you use that technique, in purple you see that when you silence those dopamine neurons, you prevent ketamine activating dopamine synthesis. So that tells us that it does require this aspect of the circuit, but upstream of that, we think that it's actually a GABAergic dysfunction that dysregulates the circuit. And using the same chemogenetic technique, we were able to manipulate those GABAergic interneurons to show that, again, you can block the effect of ketamine. So this gives us a new target for treatment. And actually, we've got some compounds now that are coming through that are able to manipulate these GABAergic interneurons. They've got some snappy names like Compound X. Perhaps we'll get a better name shortly. But here you see that there's evidence that we can use these compounds that selectively target these GABAergic interneurons to, again, block the effect of ketamine. So here I think we've shown you a translational model that can be used to develop new drugs that recreates what you see in patients using exactly the same measure. So perhaps we've got the chance to break out of this Groundhog Days cycle. So if they ever do remake Groundhog Days and do a sequel, I'm hoping there will be a scene in it where Bill Murray's character says something like this to the groundhog: "Hey Phil, there's a hope we might escape the Groundhog Days of D2-based antipsychotics." Thank you very much for your attention.
Well, thanks. You might have noticed I'm not Ian Everall. Ian is on his way to China, so he apologises he can't be finishing off today. My name is Matthew Hotoff. I'm Vice Dean for Research at the IoPPN. And I think we've just had the most amazingly kind of rich, diverse, interesting afternoon. And I mean, part of the point about having Parade of Stars is that you find out about work which is going on in a department neighbouring yours, maybe your own department, maybe even on the corridor you reside on, which you didn't know about before. And I've certainly learnt a huge amount. I've learned a lot, actually, about science I just don't know about. So, the metaphors are wonderful, aren't they? The use of metaphor in presenting the balsamic vinegar and olive oil to explain protein phase transitioning, which I knew nothing about. Will mean it sticks with me, and of course the wonderful murder mystery we heard about from Kinga, is magical. But it has spanned from those very basic, fundamental science descriptions through to really important public health applications, the impact of sitting too long. My God, don't we all do that too much? And also how difficult it is to implement something which seems like common sense into practice, and that, of course, is absolutely critical. We've heard about translational research, the Groundhog Day of D2 receptors. We've heard about epidemiology and risk factors and the things which matter to our patients and our populations. So it's been incredibly rich. I think the other thing which I've picked up on is not only there's this sort of diversity in terms of the topic which we've been having, and the diversity in terms of the career stages of the participants from pre-doc all the way through to professors, but also actually the wonderful diversity, the internationalism of this organisation. And we didn't ask people to put up a flag when they were speaking, but my God, there were a lot of different nationalities represented, and long may that last well beyond the ghastly Brexit. So I've had a wonderful afternoon. These things don't happen by accident or happenstance. They happen because people do a lot of work. And I just wanted to acknowledge our amazing comms team, led by Louise, but Robin, Abby, and Hannah as well. Please come up and receive a little something from us. Here you are.
I think Hannah might be in the canteen.
Hannah's in the canteen, right? Even more. Robin, you don't escape the flowers. And, well, thank you for everything you've done. And the timekeeping, which was terrifying to be sitting behind, because I kept seeing the notices going up and wondering, had the speakers spotted that they were due to leave? These things don't happen by accident. They also happen because of the work of the Research and Innovation Committee, the RIC. And the RIC is a very, it's a great organisation because it actually brings together people from across the IoPPN every department's represented, and there's a lot of different career stages represented. I think every division's represented, I should say. But there are a lot of different career stages. It's very much built in that this is a living committee, and it's a committee which does stuff, including this. And the committee's been very ably chaired for the last 3 years by Talia and Carmine. Talia has kindly agreed to carry on, but Carmine is standing down after his first 3-year term, and we're immensely grateful to you, Carmine. You have done a number of things. One of them is to diversify Parade of Stars, which when it first was established was a lot of professors, and actually it's so much better and richer for being for representing career stages across, people across the career stages. Carmine has also run the Independent Researcher Award competition. I know from people in my own group who've received these how important that is. It's often the first award you get. You're a year or two after your PhD, you're not quite sure where you're going, and getting one of these independent research awards, even if it's not a great deal of money, is just a hugely important kind of transition for people. It makes a big difference, and Carmine has been instrumental in making all that happen. And he's also done a lot of kind of things behind the scenes which matter to people in terms of careers, drafting procedures regarding honorary titles, and all of those things. A lot of other very unglamorous stuff, like turning up at the research committees in King's Central, or the health schools research committees, or the King's Health Partners research committees, jobs I now find myself doing as Vice Dean Research. So Carmine, thank you very much, and please come up and receive this little gift. And now I think there is drinks. There are drinks. Where are they? In the canteen. Excellent. So please make your way there.